Okay, now I'm recording. So, the first thing we have to do is, since we just got back from the weekend, uh, and we need to move on to our next section on different patterns of inheritance, let's first review the pattern of inheritance that we've learned so far. Okay? So we've learned of the pattern of simple dominance, right? We are aware of one pattern, and that is simply you have two alleles that make up a single gene which controls a trait, right? And one of those alleles is dominant over the other. And if that is the case, the dominant gene will be expressed. Uh, let's go ahead and just get into the nitty gritty of it. So I'll erase this. We are familiar with genotypes. Okay, we're familiar with genotypes. We know that genotypes are simply combinations of alleles. There are homozygous genotypes where the alleles are identical. They can be either both dominant or they can be both recessive. Let me zoom in a little bit. We also know that sometimes we can have a heterozygous genotype where the alleles are different. So you can have a dominant and a recessive allele together. And uh, taking what we know from patterns of simple dominance, we know that in this particular case, when we have a dominant and a recessive allele together, the dominant trait will be expressed in that organism. Uh, and the expression of that trait is called the phenotype. Remember, a genotype is just simply the combination of alleles. And the phenotype is the actual trait that is expressed and that we can observe in our individual organism. Okay, so let's go back to our initial diagrams where we looked at two tall plants with heterozygous genotypes. Remember that the alleles separate during the formation of sex cells, so each sex cell will only have one allele, which then has a probability of being joined with the other parent's allele during fertilization, and that'll result in a variety of different genotypes. Right? We get homozygous genotypes for dominant traits. We get two heterozygous genotypes, and we get one homozygous recessive genotype. Uh, and the different phenotypes are only two. We have tall or short. Those are our two possibilities. But we can see here that the probability of getting a tall phenotype is 3 out of every 4. For every 4 plants, approximately 3 of them should be tall in the offspring of this generation. What else do we need to cover? No, so we talked about simple dominance, which is what we're familiar with right now. I just went over genotype and phenotype. And now we need to discuss the other patterns of inheritance because simple dominance is not the only pattern of inheritance that we observe. And this principle of dominance, where a dominant allele is expressed over a recessive allele, is not always the case. There's many funky things happening with genetics, and especially as you investigate more and more complex organisms, you'll find that the mechanisms in the genetics that control those traits in those complex organisms also have some interesting characteristics and qualities. Why is too tall heterozygous? Uh, Let me explain to you why two of these tall plants are heterozygous. Let's do a Punnett square. Boom. We'll do this right here. Get a line going straight down the middle, and a line right here. So remember these Punnett squares that we did? Uh, I'm going to show you why exactly our two plants that are heterozygous are tall. Remember, Bryce, that tallness is dominant in pea plants, and that's why we use a capital T to symbolize that allele. Okay? It's dominant, meaning that it doesn't matter what combination it's in, what genotype that you find a dominant tall allele in. If an organism has this allele, it will be tall, right? We know this because tallness is dominant. Just gonna put dom here. And shortness is recessive. So if a short plant, well, a plant can only express its shortness if there is no dominant allele present. Now the parents for this F1 cross of pea plants, remember, they're both heterozygous, okay? So we're going to fill out their genotypes along the top and the left side of this particular cell. Let me go ahead and delete this. So here we go. That's parent 1. but these alleles are referring to parent 1, so we'll just put P1 here for parent 1, and then parent 2 is going to be on the left side. And again, it's also heterozygous, so it has a dominant and a recessive T. And then we fill this table out as usual. So again, my rules are I start with the dominant alleles first, and I just fill them in wherever they appear. So I'm going to fill the top ones down. I'm moving down. And now I'm going to move from left to right, going across. Now that I'm done with the dominant alleles, I'll do the recessive alleles. So I'm gonna start with the top. I'm gonna just fill this in, moving down. And now I'm gonna do the one on the left side, moving from left to right. Just going across here, and there we go. Now, you ask, why are these heterozygous genotypes? Why do they result in a tall plant phenotype? Well, which one's dominant again? It's because the tallness allele is dominant over the recessive allele. Despite carrying the allele that results in a plant being short, the dominant tall plant prevents the recessive allele from being expressed. Okay, so I know it's not centered. I know it's not centered. So that's why our heterozygous plants here are tall. And again, we can see that this is a heterozygous genotype. These alleles are not identical. You have a dominant and a recessive allele. So this is a heterozygous genotype, and because tallness is dominant, that's why our heterozygous plants are tall. Hopefully that helps you out a little bit. But let's get back to the matter at hand. We were talking about other patterns of inheritance, so let's jump right in. Now, I'd like to preface this whole lesson. Oh gosh, please! I can't answer this phone right now. I have to go. I'm sorry, I lost my train of thought. All right, let's just jump right into these different patterns of inheritance. So, let's start off with codominance here first. This is the first alternate pattern of inheritance that we can see exhibited in many different organisms, actually. So, when we have an organism that has a gene in which the alleles are not dominant over each other, we usually get a pattern of inheritance known as codominance. The example the book gives you is with chickens, okay? So in certain kinds of chickens, the allele for black feathers is codominant with the allele for white feathers. Let me highlight the rest of this. Meaning that one is not dominant over the other. So what happens? Well, since neither is dominant and they can't prevent the other one from being expressed, they both get expressed in the organism's phenotype. Okay? So let's go ahead and take a look at what this would look like in an organism. And a good example is the Sussex chicken, the black and white speckled chicken. Let me go ahead. Oh, that's a really bad image. This is the one I used from yesterday, I think. Uh-oh. I don't want to save the image. I want to open in a new tab. So here is an example of this codominance pattern of inheritance being expressed. The black feathers do not prevent the white feathers from being expressed, and vice versa. Both of them are expressed equally. You get white and black feathers, or feathers that are black and white, because of this interaction between those two alleles. And there's a way I like to refer to. Sorry about that. There's a way I like to refer to this particular pattern of expression, and I like to write that when we're talking about codominance, we're talking about equal and separate expression. Equal, separate expression. I'm going to abbreviate expression right here because I can't draw too fast. So equal, separate expression. They are co-expressing, right? And they're both being expressed equally, but they're separate, meaning that we don't get like a mixture of the colors white and black. You know, that would result in gray. This chicken here does not have any gray feathers. All right, so both of the alleles are expressed equally and separately. Okay, not mixed together, not being overpowered one way or the other. That's what we refer to codominance as. That's the kind of phenotype that we get in our organisms. Okay, so if we were to do a cross for this particular chicken, it would look a little something like this. Let me go ahead and get rid of this. Oop. So let's say we have two chickens, and we'll go ahead and put their genotypes here. So we have BB, ABB, and then we have WW. Okay, now the allele B represents black feathers, and the allele W represents white feathers. Oops. Geez, I need to get like one of those little tablets and pens so I can write this without seeming like I don't know how to write at all. Okay, so we did not, these are both uppercase alleles, and we use uppercase and lowercase in genetics to indicate whether an allele is recessive or not. So we'll go ahead and, erase this, but remember both of these, neither of these alleles is dominant over the other, right? They can't overpower the other and prevent the other from being expressed. So when you breed these chickens together, okay, you'll get the genotype BW. Each one of them will be heterozygous. Okay, and if you wanted to fill this cross in to double check it, I welcome you to do that, but this is the outcome. You breed a homozygous black chicken with a homozygous white chicken, all of these offspring will be born with black and white feathers, an equal separate expression like we see in our image right here. Okay, this is what the heterozygous phenotype looks like when you're talking about a codominant pattern of inheritance. Okay, let's move on to the next one. And most people, when we're doing this unit, get really confused between codominance and incomplete dominance. And I'd like to sort of make sure that we're familiar and comfortable in distinguishing between the two. So there are some instances in which a dominant allele is not completely dominant, meaning it doesn't completely 100% prevent another allele from being expressed. And a great example of this is our 4 o'clock flowers. 4 o'clock flowers have some alleles that control the color of their petals. You have the R allele, which results in the flower's petals being expressed as red. And you have the W allele, which together in a WW genotype will result in the petals of this flower being expressed as white. Okay? Now, it is unclear which one is dominant or dominant or which one is not dominant, but regardless, whichever one is dominant, it's not completely dominant, meaning the other allele is expressed. However, what makes this different from codominance is that it is a mixed expression. It's not a separate expression, right, the way that complete dominance or codominance is, right? Our chicken had an equal expression of black and white feathers with no mixing. If those two colors mix together, you get gray feathers. We don't see that in our speckled Sussex chickens, okay? But we see this in 4 o'clock flowers. So if you turn your attention up here again, we have two homozygous parents, one with two red petal alleles and one with two white petal alleles. And the offspring are all going to have a heterozygous genotype RW. But because this pattern of inheritance is incompletely dominant, the result is these two colors mix together and result in pink. When you take red and white together, you get a pink color. And that's what happens in these plants. So a great way to remember incomplete dominance is that this is a mixed expression. Whereas our speckled chicken was a separate expression. The different black and white feathers were expressed separately. In this case, our petal color is a mixed expression of red petals and white petals, which gives us pink. Okay? So incomplete dominance is a mixed expression. Codominance is a separate expression. Both are equal. Both the alleles are expressed equally. It just depends on whether they're expressed separately, as in our chicken with the black and white feathers, or if they are expressed in a mixed format, meaning we take our red petals and our white petals. When you combine those colors together, you get pink. So that is the difference between those two. Okay, that's probably the trickiest part of this section, is just distinguishing between those two particular patterns of inheritance. All right. Okay. Let's go ahead and talk about a couple more patterns of inheritance. And we're going to stop at multiple alleles. Now, so far, I've only introduced you guys to genes that are controlled by two alleles, of which there are two versions. Okay, so going back to our pea plants, remember our pea plants had two different alleles for the plant height gene. Okay, two different alleles for the plant height gene. However, that's for most organisms. There's not just two options to choose from. There's more. There's more options to choose from. And if we were to write out these alleles separately, I mean, let's say we have a dominant allele and then a recessive allele, but we could have another dominant allele, which we'd label as T1, or a third allele, which we label as T2. Whenever we have multiple alleles, that's usually how scientists like to record them and keep track of them visually, is by adding those little superscripts to their alleles. You're not going to have to worry about that on your test, but I'm just letting you know in case you were interested. There are more than two possible alleles for many genes, especially in complex organisms. For our pea plants, they were very simple. Their genetics were very simple. They only had pretty much two options, or two different alleles for most of their traits. But again, that's not the case for every single organism. When you're looking at rabbits, rabbits have 5 different alleles. It says 4 here. It says 4 right here. But in reality, there are 5 different alleles that control rabbits' coat color. So it's possible. It's possible to get many more combinations when you have 4 different alleles. And how do I know this? Well, it's simple. Let's go ahead and look at. Oops, hold on. If we're talking about just 2 different alleles, sorry guys, I got a little carried away. If we're talking about 2 different alleles, we have 3 possible arrangements of this particular of genotypes with these alleles. We have homozygous dominant, we have heterozygous, and we have homozygous recessive. With only 2 alleles, we have 3 possible combinations. But when we have multiple alleles, it really opens up the different combinations that we can use. So if we were talking about 4 different alleles, I mean, you're talking about an exponential increase in different genotypes, which may result in a lot of various phenotypes as well. And so that's why we see lots of different expressions in the fur color of rabbits. You know, you have rabbits that are completely white, rabbits that are how do I say this? Painted, meaning they have, you know, sections that are different colors. We have rabbits that are gray, brown, chestnut in color, so on and so forth. And that's because there are more options for alleles than just 2. Okay. I wanted to show. Nah, we're okay. Another example of a gene that has multiple alleles would be blood type. So actually, we could probably do that. I'll do an activity for our human blood type later. I'll post it in the Google Classroom probably on Thursday. All right, so we've talked about codominance, where we have a separate but equal expression. We talked about incomplete dominance, which is a mixed but equal expression. We talked about genes that have more than 2 alleles, so multiple allele genes. And we also gave some examples for each of those different patterns of inheritance. Now we're moving on to polygenic traits. Polygenic, poly, the first part of this word meaning many, and genic relating to the genes. So polygenic, it means many genes. Oops, let me erase this. So let me give you guys some examples of organisms that have polygenic traits. So for example, the book gives you fruit flies. fruit flies have 3 genes that control the pigment in their eyes. 3 different genes. Meaning, if I'm going to illustrate this right here. Let me go back here. And I like to take this opportunity to restate to you the difference between a gene and allele. So let's go ahead and draw it out. So let's say we have any particular gene here. This is a gene. Okay. Now a gene is going to be a combination of alleles. So we'll just say X, X, right? A gene is when you have a complete set of genetic information, and that includes having alleles from each parent. Now remember, the gene is comprised of 2 alleles. So we can have 2 different alleles here, X and X. And that's how we distinguish between genes and alleles. The genes are just combinations of different alleles, okay? Now we know that if we were going to, let's say there's 2 different versions. There's an X allele and a Y allele, right? And each parent has a chance of contributing one of those alleles to their offspring to create a genotype. Now, we can track that probability, and the probability of a specific genotype being reached with a single gene is much easier. All we have to do is take 1/2 and multiply it by 1/2, right? So there's a 1/2 chance of there's a 1/2 chance for either of these specific alleles to be passed down to offspring, right? And so we can calculate the probability by multiplying those 2 together to figure out which combination we would get. Now, what happens if there's more than 1 gene, right? Well, let's say we have 3 genes. Gene 2, and then we'll do gene 3. And each of those genes will also be controlled by 2 alleles, right? And each of those alleles will have a 1/2 chance of being passed down to their offspring. And if you wanted to get a very specific combination, an incredibly specific combination of alleles, then you would have to literally multiply all this together, right? If you want a very specific combination of alleles from each parent, you will multiply this all together. And let's do this really quickly and see what happens, right? Well, let's just look at the first gene. So let's say we're just focusing on what's the probability of getting a specific combination and with just 1 gene. Well, again, it's just 1/2. 0.5 times 2. Oh, wait, no, not times 2. 0.5 times 0.5. Come on, Mr. Holden. So you get a 0.25, which equals a 25% chance, which also equals 1/4, right? If you wanted to aim for a specific combination of genes, you would have a 1 in 4 chance of doing so. However, now we're looking at 3 genes, and if we wanted a very specific a phenotype, and we needed to be very careful about which genotype we selected, we were going for a specific genotype. Now we have to keep multiplying to figure out what's the percent chance of breeding the very specific genotype that you want. So let's do that right now. Okay, we're going to continue multiplying by 0.5. We got to do how many more times? 4 more times. So multiply 0.5 times 0.5 times 0.5 times 0.5 equals 0.015625. I'll multiply this by 100 to give us the percentage. So a 1.5625% chance of getting a very specific combination of genes. Now, let's say there's also some exceptions. Like, this is probably not the true percentage, because it's possible that you don't get the gene that you wanted for one parent, but then you got the gene that you wanted from another. So you have to account for those exceptions as well. But for the most part, you can see that if I had 3 genes controlling a single trait, I'd have a lot more options and a lot lower probability of getting a very specific genotype in this situation. Okay, so that is a polygenic trait. And other examples, I mean, humans have many polygenic traits. The first being, first and foremost, being eye color and skin color. Those are controlled by, well, skin color is controlled by 4 different genes. So again, if I'm looking back at my paint diagram, I'd have to add another gene in here to get a specific genotype for skin color. which is going to be even lower likely a less likely probability of getting a specific genotype when you have 4 genes, right? So what does this mean? This means that the more genes control a trait, the more variety you have. We see a lot of variety in skin color. We see a lot of variety in eye color as well. And I remember Natalia asking me about a week ago, well, we see instances of parents that have seemingly dominant traits, but then their kids are born with recessive traits. I would chalk it up to being a polygenic trait, and perhaps there's some other patterns of inheritance working as well. So we can have a trait that is controlled by multiple genes, and each of those genes can display their own patterns of inheritance as well. Like we can have codominance and incomplete dominance in multiple genes of a polygenic trait, and that might give us even more variability as well. So that pretty much does it for those different patterns of inheritances. We're stopping at polygenic traits. However, we have a few more things to go into before we're done. But let me go back to the chat and see what questions you guys have been asking. Second screen. I have a quick question. Class because we have Google. Yeah, I'll talk about Google Suites at the very end. It's not center. There are blue rabbits. How is multi and poly different? Okay. Let me go back and explain multiple alleles in polygenic traits. I'll do another paint diagram. I gotta start from scratch. Okay, so you have a trait, right? Let's go ahead and just write out trait here. Now, a trait is controlled by a gene or genes, okay? So I'm just going to do a single gene here. I'll put a gene right there. You have a gene that controls a trait, okay? And the gene is itself controlled by 2 alleles. Now, in the lessons we discussed so far, the pea plants that Mendel was working with typically only have 2 possible alleles for their different genotypes and phenotypes. So they'll have allele A and allele B, right? However, many genes actually have more than 2 possible alleles. So in pea plants, even though there's only 2 possible alleles A and B, depending on which gene you're looking at, there's also probably an allele C. Well, in other organisms, there are often more than 2 alleles. So there's probably an allele C that exists. There's probably an allele D that exists. And these aren't how we symbolize those specific alleles, but I'm just showing you for your understanding. And then maybe there's another allele that's possible. Okay, allele E. And this is a great example of a multiple allele trait, specifically in rabbit fur color. So rabbits have 5 different alleles that can express different colors of fur. And so a parent may have any combination of these 5. It might have A and B. It might have A and C. It might have E and D. I mean, the possibilities are much greater than if you just had 2 different alleles. Does that see what I mean? And the difference between multiple alleles and multiple genes is, in the case, instead of having multiple different possible alleles, in this specific instance, when we're talking about polygenic traits, we have multiple genes, each with their own alleles, that also control a trait. So these are really bad arrows. I'll just do lines. All right, delete this. Delete this. So you have multiple genes that control a trait, whereas in multiple allele traits you have more than 2 different alleles that you can make possible combinations and genotypes with. Does that make sense, Bryce? Did I answer your question effectively? So again, this right here, when you have multiple genes that control a trait, this is a polygenic trait. Okay. However, when you have a gene that has more than 2 possible alleles that can make up its genotype, then you're looking at a multiple allele trait. So that is the difference there. Okay? You think what, Bryce? Think you got it? All right, I'll give you some time. Okay, let's go back and wrap up this particular section, and we're going to talk about environmental stimuli on genes. And a great example for this your book gives you is the western white butterfly, species name Pontia occidentalis. Um, the western white butterfly is a native species. Oh God, I don't want to get rid of this note. It's a native species to the western part of the United States. And some interesting things happen, or some interesting characteristics are displayed and expressed depending on what time of year that specific butterfly is born in. Okay. What does this mean? What does this mean in terms of what we know about genetics so far? Well, it shows us that genes are not just the phenotype of an organism is not simply controlled by the genotype, right? There are other factors that can affect the phenotype. Remember, the phenotype is the appearance, right? That is whatever characteristic is expressed depending on the genes. But what the western white butterfly shows us is that there's also influences by the environment that control the phenotype, not just the genes itself. If you're thinking back to Jean-Baptiste Lamarck and his idea that an organism can change during its lifetime, it's similar in that regard. But I think the main way that we move away from Lamarck's ideas when we're talking about this specific concept is that it's not necessarily controlled directly by the organism. So, how does the environment influence the western white butterfly? Well, let me explain it to you. Basically, they will mate and will lay eggs and will reproduce in different seasons. So they'll reproduce in the summer, they'll reproduce in the fall, they'll reproduce in the spring. Um, and what happens is we get different colors depending on what season, well, sorry, different wing colors of these butterflies depending on what season they are born in. So this is interesting. Individuals that might have completely identical genotypes will have different phenotypes or different appearances depending on the time of year that they're born. What is causing this? Okay, what is causing this? Well, it's it's been determined, or there's evidence to support that temperature has an effect on the color of the wings, and not only temperature, but also perhaps the amount of light that the eggs or the larva or the juvenile butterfly, the adult butterfly is exposed to. So temperature and light may play a role in what pigment or how much pigment is produced in these butterflies' wings. And let me just if this is confusing to you for whatever reason, let me ask you a question. What happens when you go in the sun? If you don't get sunburned, your skin definitely darkens. You get tan. Your cells begin to produce more pigments to protect your skin from electromagnetic, basically UV radiation protects you from that light, so the cells don't get damaged. So the light itself is a stimulus to activate your genes that produce pigment. Okay, so the same thing happens in butterflies. It's probably it probably has something to do with the shorter days in springtime and the longer days in the summer. So, for instance, in the springtime, the days are much shorter, right? If you guys notice right now, we're entering summer and the days are much longer. I mean, the sun's out at 6 and it's down at 7:30. So we have really long days right now because of the angle of the sun at this time of year. Whereas in springtime, we still have a low angle of the sun in the sky, so the sun hits, the sun is up for much less time than it is during, summer season. Okay. Now, depending on whether there's light or not, or depending on the temperature, the genes that control pigment can either activate or deactivate. It says right here in your paragraph that butterflies hatching in the shorter days of springtime had more pigment in their wings, whereas in the summer they had less pigment in their wings. It's possible that the genes that control pigment are activated in the dark, which is why The springtime butterflies had darker pigment. When there's less light, or maybe it's a lower temperature too, the genes for producing pigment get activated. And we see this a lot too. I mean, this happens every single day when you guys eat. When you guys eat, your brain releases a neurotransmitter leptin to indicate to your body that you're full, indicate to your body and your brain that you're full. I don't need to eat anymore. Is that all that neurotransmitter does? No. Well, that neurotransmitter will tell your cells to begin producing enzymes and other molecules needed for digestion. So the presence of leptin in your body will trigger your cells to start making all the tools needed to digest the food you just ate, okay? And in order for them to be produced, the genes that control their production have to be activated. So your body is signaling to your cells to activate the genes to produce digestive molecules and materials, okay? The same thing happens, like again, when you're in the sunlight. When you're in the sunlight, the light itself activates your genes for producing pigment, and your cells begin to do that. Okay, genes can be activated or deactivated depending on environmental stimuli. So, despite everything we've talked about, when we discussed how just the phenotypes and the characteristics we see are related to specific genotypes, it's possible that the environment also plays a role in whether certain genes get activated or not. Okay, so keep that in mind moving forward. It's possible to have two individuals with the same genotype but vastly different phenotypes depending on the environmental stimuli that have been exposed to each individual. Okay? Yeah, that's why the spring butterflies look darker. It makes sense for them to be darker. In the lower light levels, if they were bright colored, they'd probably be easy to, their contrast would make them stand out. But during the springtime, they're probably going to be darker as a way to avoid being caught by predators. So maybe the cold temperature or the low amount of light results in the gene for producing pigment to be activated. And well, let's see. Let me try to draw this out. I'll go down here. So in the summertime, right, we have longer days. Much longer days than if we were in the springtime. So we'll have a little short day right here. It's possible that during the daytime, I'll put daytime right here. It's possible that during the daytime, the light or the temperature itself deactivates the gene for pigment production. So that gene can only be activated at a lower temperature, or it only activates when there's no light present. So because the days are shorter in spring, there's more time where there's no light, or there's just a lower temperature in general, which causes the gene to produce pigment to activate. However, in the summer, since the days are longer, it's warmer, there's much more light available for a longer part of the day. The genes for making those pigments are probably deactivated during this time and only activate when there's no light or when the temperature drops. So that's probably why we see this difference between the pigments of those butterflies between seasons. Okay? That pretty much concludes our lesson. We are done with 11.3. I'm going to go ahead and go to my Google Classroom right now. If you guys go to the classwork tab, you'll see the 11.3 worksheet here as a PDF form. So please get started on that. That'll be due Thursday for you guys. I've already posted it in the description here. So get started on that as soon as we're done. One thing I also wanted to talk about today, since someone asked, excuse me again. Someone asked about Google Suites and what's going to happen with that. Your homeroom teachers should be reaching out to you with a brand new email that is linked to the Village Glen domain and will allow you to access all the Google Suite options. The school paid, well, I don't know if the school paid, but the district paid a lot of money for us to implement this and use it. So what's going to happen in this next week is your teacher's going to email you your new passwords and email addresses. You'll have to log in, change your password, and then accept invitations to your new Google Classrooms, okay? So you guys, we're not going to be on this Google Classroom anymore. It'll be a new one, but it'll probably be the same format. It'll still have the same sections and everything. It's just allow us to use more features. For instance, we won't have to do YouTube streams anymore because we'll be able to fit everyone in a Google Hangouts, and I'll be able to mute people, and I'll have admin privileges and stuff like that. So we should be able to you know work with those, but I don't think they're that necessary. So please check your emails, your current email that you're using right now. Check this email for your new email, for your Google Suites email, okay? And then by Monday, everyone should have it all figured out. So I'll send you guys invitations to my classroom once I set it up on G Suites, and you guys can join it ahead of time. But I'm not going to officially start teaching and posting things to that page until Monday. All right. I know you like your email, but the problem is, I think one of the problems is when you guys use your own emails like you're using here. I know who you are because I've seen those usernames and stuff, and I'm familiar with who has which email name or profile name or whatever. But you know, other teacher aides might not be able to keep track of it as well. It just makes it probably a little easier if we all use very professional emails that display our first and last name. Tuesday, you mean? What happened? Wait, Tuesday? What do you mean? Oh yeah, sorry, Tuesday. Yeah, we'll be in a different Google Classroom. What I mean, we're gonna have a different classroom. I mean that this Google Classroom page that we have all grown so fond of, and we are used to, we are switching to a new Google Classroom because we just got access to G Suites. It's gonna look very similar, pretty much identical. I'll have all the third and fourth period students in that classroom, but all the classwork will be gone. I'll probably have to repost that. I will only repost the assignments that are for the fourth quarter. So some of these assignments are from the third quarter, which is already over. I'll post those into G Suites. So uh, everyone, it's not a Gmail. It is a Gmail email. It just has a domain associated with it. So it'll be bsatnick@villageglen.org. I know it's not gmail.com. It's not at gmail.com, but that villageglen.org is a domain hosted by Google Suites. So you guys have email addresses with that domain, meaning you're allowed all of the access to Google Suites as long as you use that email. Okay? Anakin will help us. We are killing young. He will save us by killing us. No, there is no slaying of younglings in my class. I know we are in distance learning and you guys are home, but this is still my class and my time, and there are no slaying of any younglings. Okay, that's one of my number one rules. Younglings are not to be slain. Okay. All right, I'm gonna stop the recording right here.